Good afternoon. Today I want to talk about attachment and entry of viruses into cells. We're going to talk about how we get the genome out of this poliovirus particle. Now, last time I didn't want to get the genome out. Today I'm going to do that. As I remember, this is the whole genome, 7,440 nucleotides, and it's in the right sequence. In fact, you can see at the end here, it's all red. That's the poly A tail, so A is the red beads. And it's very long, and it's very hard for me to get out. Now, I've already taken a few seconds to do this. Of course, in the cell, it would already be out. It's out. The genome is out of the particle in milliseconds. And I'm not even going to bother because it's all tangled up in here. I can't get it out. But what you can see, this is the 5' end, and the beader put a bigger bead on there. That's a protein linked to the 5' end. So the amazing thing you can learn just from watching this is that the way nucleic acids are packaged inside virus particles is very special because they have to get out very quickly. Now, there's a little hole here. One of the subunits has been removed to allow me to get this genome out. But in fact, in reality, the opening is even smaller. So somehow that whole 7,440 nucleotide genome gets out through a hole in milliseconds. And of course, I have a bigger opening. It's confounding me. So there you go. There's the magic. There it is. It's finally out. That's the whole thing. I don't know. This is about 10 meters long in reality. It's all tangled up. Of course, is the RNA actually straight like this? No, it's all coiled up and secondary structured and so forth. Now, getting it back in would take me even longer. So we're not going to do that. That's actually the subject for another time anyway. Today I want to talk about how we make viruses unstable. Remember, we talked about them being metastable last time. They're very stable as they go from cell to cell and from host to host. But at some point in the cell, they have to become unstable. They have to give up the genome. They have to become less stable. They have to let that genome out, as I just did for poliovirus. So we're going to talk about that, how that happens. Now remember, viruses are obligate intracellular parasites. They have to get inside of a cell in order to replicate. Sitting on the outside is not good enough. They have to get their nucleic acid in the cytoplasm or in the nucleus and replicate. And here's an infectious cycle just to remind you, we have viruses getting into cells and doing all sorts of molecular activities that we'll talk about in this course. But they're too big to simply diffuse across the plasma membrane of the cell. They cannot knock into a cell and diffuse in. Too big, not happening. So they're taken up by specific pathways, and that's part of what we're going to talk about today. And you see here is a virus binding to a cell receptor, and that's part of what allows viruses into cells. So how does a virus find the right cell? As you'll see later, when you're infected with a given virus, not every cell within you is infected. And every virus has a different way of getting in and of spreading in different fluids. Some viruses are respiratory acquired, and they remain in your respiratory tract. Others get into your blood, say by a mosquito vector, and can travel basically anywhere. So a virus that's in the blood has a ticket to the world that is your body. How does it find the right cells? Because most viruses don't replicate in every cell. Well, it turns out that viruses bump around a lot. In your blood, they're always bumping into cells, and most of them don't lead to infections. And a lot of those early interactions are simply electrostatic. The virus bumps in, sticks briefly to the cell, then falls off. But if there is a specific receptor molecule on the cell surface for the virus, then the virus will or may attach and get into the cell that way. So I want to talk first a little bit about receptors for viruses, and then the pathway used for entry into the cell. Now, sometimes there's one receptor on the cell surface for the virus. Sometimes there's more than one, and the situation becomes complex, as you'll see in a moment. Then, after the initial binding, the genome gets into the cell. And there are multiple mechanisms for that I want to talk about with you today. Now, cell receptors for viruses. These are proteins or molecules on the cell surface that are required for entry. They're needed for All the animal viruses that we talk about, they're needed for bacteriophages. They're not needed for viruses of fungi. These viruses typically don't have extracellular phases. They divide inside of the cell, and then as the cell divides, they go into the new cell. You have to worry about the outside world. They don't have to worry about getting back into cells. Plant viruses also do not bind to receptors on the cell surface. They are introduced into plants by vectors, insects, worms, or mechanical damage caused by farm equipment. That introduces them into the cells, so we don't need receptors for those viruses. But all the other viruses we talk about require a receptor. And this is a relatively recently developed field. As recently as 1985, we only knew one virus receptor, which happened to be a sugar, sialic acid for influenza virus. But that situation changed rapidly with the development of several technologies, monoclonal antibodies, allowing you to specifically identify cell surface proteins that virus is attached to, and recombinant DNA technology. This has really changed our understanding of many fields, especially receptor biology. Now, at the top of this slide is a picture of a generic plasma membrane in a eukaryotic cell. As you know, it is a lipid bilayer that keeps things out and keeps things in the cell, and it has to have pumps and so forth to get things in and out. Embedded in that lipid bilayer are various proteins. There are integral membrane proteins, where a portion of the protein traverses the membrane, and then there's an extracellular and an intracellular part. There are indirectly anchored proteins, where a second protein non-covalently associates with an integral membrane protein. There are lipid-anchored proteins that don't traverse the membrane. They're attached to the leaflet by lipid moieties. And in addition, of course, some of these proteins are glycosylated. We call them glycoproteins, and that plays important roles in virus attachment. Now, the surface of the cell has lots of different proteins in it, all of which have roles for the cell. And one thing I want you to understand at the onset is that none of these proteins exist for viruses, okay? None of them are there so that a virus can get in. Viruses have evolved to be able to bind them and get into cells, but these proteins have normal roles in taking up material from the extracellular milieu. They bind materials, they traffic into the cell, and those pathways have been evolutionarily adapted by viruses. So they do not exist for virus entry. It's an accident that this happens. Now, at the bottom here is a representative selection of some different receptors for viruses that we know about. From the left to the right, you can see a variety of molecules, both transmembrane proteins and lipid-anchored proteins like the GM1 ganglioside. These are monomers, some of them are multimers, and they have a variety of cell functions. There are proteins involved in immune functions, for example, CD4, CCR5. There are proteins involved in cell-cell adhesion, but they're on this slide because each of these proteins is a receptor for one or more viruses. And the viruses are listed at the top here. And you can see here's these two proteins, CD4 and CCR5, or CXCR4, are receptors for HIV-1. That particular virus needs two receptors to get into cells. Adenoviruses bind to these receptors down here. And you see there are multiple virus names at the top here. They're all abbreviated, but here rhinoviruses bind to LDL-like receptor. So any surface protein can serve as a receptor as long as it allows virus entry. And we'll see how that works in a moment. But remember, all of these proteins have some role in the cell. So it's not a good strategy to try and prevent virus infections to remove this. You know, in years to come, we'll be able to edit humans and take out genes that we think we don't like. You shouldn't take out a gene for a virus receptor because it's got some other function that you're going to interfere with. In fact, in general, we probably can't take genes out of us because of that reason. Many viruses bind to one receptor, some require two. And an interesting situation has recently been described for Zika virus, which is shown here. You know, this virus emerged mid-2015, and everybody seemed to jump on it and work on it, and a ton of papers have come out in the past year, and I have to admit that we've also started working on it as well. And a paper just came out in the last 2 weeks showing that Zika virus entry, at least in some cell types, not others, needs 2 proteins. One, a soluble protein called Gas6, which actually binds the virus. And Gas6 is a natural ligand of a second membrane-bound protein called AXL, AXL. So together, this allows virus uptake into the cell. This is a kind of an unusual situation, but it works because AXL is taken up into the cell. GAS is a natural ligand of AXL. So there can be lots of different situations with respect to receptor-mediated entry. In addition, different viruses can bind the same receptor. So here are some examples of that. We have adenovirus and Coxsackie virus. So here's adenovirus with the fibers at the 5-fold axes. Coxsackie virus is a virus very much like poliovirus. It's called a picornavirus. They look very different. They're both icosahedrally built, but different attachment strategies. They have a common primary receptor, which creatively is called the Coxsackie virus adenovirus receptor, or CAR. And Coxsackie virus, by the way, is named after a town in upstate New York, Coxsackie, New York. Exit 21B on the thruway. You've been to Coxsackie? Yeah. The virus was isolated from there in the 40s. And once I was driving past, and I said, I have to get a picture of this for my classes. And I held my camera up, which you shouldn't do, and that's it. It wasn't a phone camera. It was a big 35 millimeter camera. But I'm really proud to have Coxsackie exit on the program. A herpes virus, pseudorabies virus. This is a herpes virus here, the envelope virus, pretty large. It binds the same receptor as human poliovirus. So these are viruses from different families binding the same receptor molecule. Sometimes viruses in the same family bind different receptors. For example, rhinoviruses, common cold viruses, at least 3 different receptors are responsible for rhinovirus entry. Retroviruses have at least 16 different receptors. And one virus can bind multiple receptors depending on the cell type. Here's a herpes virus that can bind to a variety of different cell surface proteins. It's sometimes the case that the same receptor functions in all different tissues for a virus. So poliovirus, for example, infects the gut and then the nervous system. It seems to bind the same receptor in both areas. But Zika virus, the receptor I showed you earlier, this combination of Gas6 and AXL seems to be important in certain cells like astrocytes and glial cells in the brain, but not neurons. Neurons seem to be independent. Entry of Zika into neurons seem to be independent of AXL. So you can't assume that it's one receptor in all tissues. It can vary. So let's talk about how the viruses attach first to these receptor proteins. There are a couple of different strategies that are used I want to talk about. First is illustrated on the left. Now, when you think about an icosahedral virus like poliovirus, you're wondering first, how would this attach to a cell surface receptor? Well, it turns out that poliovirus has a corrugated surface. You don't see it very well in this plastic model. This model is from the 80s, before the 3-dimensional structure of the virus was worked out. But on the upper left there is the model based on the X-ray crystal structure of the virus, and what we know from that is that there are depressions on the surface in which the receptors bind. So on the lower left is a cartoon of the virus, or part of the virus, shown in a cutaway form. And here's the 5-fold axis of symmetry shown by the pentagon. And then here you can see a groove into which the cell receptor is fitting. Cell receptor for polio is called CD155. It binds around the 5-fold axis, and there's a groove that fits in there. You can't see it very well in this picture on the upper left, which is a cryo-EM reconstruction of the virus receptor complex. So all these gray molecules are receptors. This was made using soluble receptor molecules, so they're not membrane bound. They're added to the virus in saturating levels, so they attach to every receptor binding site on the particle. Then the cryo-EM structure was done. And you can see here is a 5-fold axis of symmetry right in the middle there. And around it are 1, 2, 3, 4, 5 copies of the receptor. And around it is the groove. Can't very well see here in this projection, but take my word, there is a depression there into which the receptors are binding. We don't think you need all the receptor sites bound in order to get infection, just a few probably at one axis of the molecule. So at least one strategy is for the receptor to fit into a groove or depression on the virus surface. But that doesn't always have to be the case. The closely related rhinovirus, shown in the upper right here, is much smoother than polio. It does have a canyon, or that groove around the five-fold axis. And here, the five-fold axis would be right there in the middle of the particle. The groove would go around it. But that picture is a reconstruction of one of the rhinoviruses bound to its receptor, which happens to be low-density lipoprotein receptor. It's a molecule important in cholesterol uptake. And that receptor is binding right on top of the five-fold axis. So there in the middle would be the five-fold axis, and you have 1, 2, 3, 4, 5 copies of LDL receptor sitting on top. And this virus has a groove around the five-fold axis, yet the receptor doesn't bind there. If you looked at this image of polio on the lower left again, here's where the poliovirus receptor binds into polio. If this were rhino, the receptor would be sitting up here on this little plateau at the five-fold axis. So an icosahedral particle can bind in receptors many ways. Now, adenovirus presents a third possibility for an icosahedral virus binding to a receptor. And that's, of course, because these viruses, not only are they built with icosahedral symmetry, you should recognize the lovely icosahedron by now, right? There's an equilateral triangular face. At each five-fold axis, there is a fiber protein sticking out with a knob at the end. And that is the part of the virus that binds the receptor. It's the very tip of the fiber, the knob it is called, that binds the cell receptor. Here on the left is the structure of that construction exploded. The penton base is the part that sits right in the icosahedral capsid. Then we have a shaft, which is actually a trimer of 3 individual polypeptides, and then the knob is at the top. And the knob contacts the cell receptor. In fact, that is what is shown at the top part here in panel B. We're showing residues of the knob in blue and red and yellow contacting the receptor protein in light blue behind. So this virus doesn't attach by having its capsid contact the receptor directly, but rather through a fiber that's sticking somewhat away from the receptor, from the virus surface. So those are 3 ways that icosahedral viruses can attach to cell receptors. Let's move to viruses with envelopes. Now, viruses with envelopes can have a helico genome inside, or it can have an icosahedral capsid, doesn't matter, but there's a membrane around it. And remember, that membrane is embedded with viral glycoproteins, and those glycoproteins are what attach to the cell receptor. So here's a diagram of influenza virus with its 8 segments of RNA. It has a membrane surrounding those RNAs. Embedded in the membrane are a couple of different proteins. The major one is called the hemagglutinin, or the HA. Then there's a less prevalent one called neuraminidase. And finally, a very rare one called the M2 ion channel. The hemagglutinin is the molecule that attaches to the cell receptor. And that's being shown here on the cell surface. This particle is engaging a number of cell receptors. It is the hemagglutinin that attaches to the cell receptor for the influenza virus. And in general, for all of the envelope viruses with glycoproteins, it is a glycoprotein that attaches to the cell receptor. Makes sense. The receptor for influenza viruses is a sugar called sialic acid. Sialic acid is present in glycoproteins. So you can have a protein, as shown here on the upper left, is the transmembrane protein. It is glycosylated. The little spheres are sugar residues that have been added to it. Sialic acid, when present in a glycoprotein, is always the terminal sugar on a sugar chain. You can see the little red arrows are pointing that out. Sialic acids are present in every tissue in your body. They have a variety of important cell functions. But it's also the sugar to which influenza virus grabs onto when it attaches to cells. There are 2 kinds of receptors for influenza viruses. Human strains typically like to bind to what we call alpha-2,6 linked sialic acids. So here on the top part of this slide are diagrams of what this means. So here's sialic acid on the left with the yellow 6-carbon ring. So this is a typical sugar ring. And it is joined to the second sugar, which here happens to be a galactose, by here at the bottom is the alpha-2,6 linkage. That's the human virus preferring sialic acid. So human viruses like to bind sialic acid when they're linked to the second sugar by this alpha-2,6 linkage. Avian strains of influenza viruses, there are lots of influenza viruses that affect all kinds of animals out there, but birds are a big reservoir of the virus. They prefer to bind alpha-2,3 linked sialic acids, and that's shown at the top here. Sialic acid linked to the second sugar by an alpha-2,3 linkage. So simply the linkage to the second sugar can make a big difference as to what kind of virus will bind to it. There's also another kind of sialic acid that is bound by the third kind of influenza virus. As we'll see later, human influenza viruses occur in three types, A, B, and C. A and B will bind N-acetyl-neurominic acid, which is up here, and the C viruses bind a slightly different sialic acid, 9-O-acetylated. Now let's look at this interaction a little more carefully. Here is a crystal structure of the influenza hemagglutinin molecule on the left. Again, derived by X-ray crystallography a number of years ago at high resolution, so we know where every atom is located in 3 dimensions. It's a membrane protein. There is a part of the protein that extends through the viral membrane, and then there is a fibrous stem made of alpha helices, and then a globular head made up of beta strands. Okay, and this is a trimer in the hemagglutinin. And when I show you a picture of influenza virus, it's a trimer in the virus. Those spikes, those individual spikes, go back to this slide, these individual hemagglutinin spikes are actually trimers. 3 monomers make them up. The sialic acid binding part of the HA is all the way at the top. Makes sense because this protein is perpendicular to the virus membrane. It's what binds the sialic acid. Sialic acid fits in a groove right at the top, and the whole top of the molecule is expanded on the right here. You can see the viral residues in blue, and there in green is the sialic acid. That's the one sugar that fits in to the pocket in the HA, and that's how the virus interacts with its cell receptor. We know exactly how these residues interact between the sugar and the individual amino acids. If you change these viral HA amino acids, you can block binding and prevent infectivity, of course. Another envelope virus that we'll talk a lot about this course is HIV-1, shown on the left. This is a retrovirus, but it's enveloped. The genome is packaged in a capsid inside, but it's surrounded by a membrane which is embedded with glycoproteins. And this glycoprotein is called variously GP120, or SU, I will be referring it to, binds to a cell receptor called CD4. In the middle is a structure of the CD4 receptor shown in brown with the glycoprotein of the virus, which is shown in red. So again, this spike on the surface of the virus binds CD4. In the middle is that co-structure of the complex. And on the right is a drawing of it so that you can see how these two interact. There's a cavity in the bottom of SU, the viral glycoprotein, into which a phenylalanine, a very specific one in the CD4 molecule, fits. And that's how the two interact, leading to virus infection. This is a virus that needs 2 receptors. We'll talk about why in a moment. All right, a question for you. Viral receptors on the cell surface. A, can bind directly to icosahedral viral capsid proteins. B, interact with glycoproteins of envelope viruses. C, can be carbohydrate or protein molecules. D, have cellular functions. E, all of the above. The answer is E. All of these things are correct, which most of you got. They can bind directly to icosahedral viral capsid proteins. I told you about polio as an example. Interact with glycoproteins of envelope viruses, influenza, HIV. Can be carbohydrates or protein molecules, absolutely. Sialic acid or protein receptor like the polio receptor, and they have cellular functions, of course. All of these are correct. And those are the important take-home messages from what I've just told you. So we've talked about binding of viruses to cell molecules. Now the important part is viruses getting into cells, because they have to put their genome there so that it can start to replicate to make more virus particles. So this is cell entry. Now cells, of course, are really good at taking up things from the extracellular milieu. They have to. They have to take in large molecules that don't diffuse across the membrane. And they have a number of processes in order to do that, which are outlined on this slide. On the left is a process called phagocytosis, where large particles are taken up by cells, typically 1 to 2 microns in size. This is a process that's used to take up pieces of dead and dying cells, and also entire bacteria, for example, but not going to concern us anymore today. On the right, the two kinds of endocytosis are what we're interested in, because these are ways that cells take up smaller materials. Endocytosis comes in two forms: macropinocytosis, where small molecules are simply taken up into vesicles at the cell surface. The membrane, pieces of the membrane move out and ruffle over material and take it into vesicles, which is then brought into the cell. And we think that some viruses get into cells this way. And then we have receptor-mediated endocytosis, where specific molecules are taken up by their ability to bind receptor molecules on the cell surface. So this is a very specific uptake mechanism. The binding of the ligand to the receptor induces the formation of vesicles, and that leads to endocytosis, uptake into vesicle, small membranous vesicles, and delivery to certain parts of the cell. So viruses get into cells by one of these two ways. An important thing to remember is that when a virus gets into the cytoplasm, it doesn't just diffuse around and get where it needs to be. The cytoplasm is really crowded. This is a wonderful drawing by David Goodsell, who makes wonderful pictures of molecules in cells and viruses too. And I divided it into 4 panels because it's too long. It starts from the top of the cell, the plasma membrane here on the upper left, and that's his rendition of plasma membrane proteins. And then below that are actin filaments, and then moving into the cell you have microtubules, of course. Moving further in, looks like we have some of the secretory apparati, the endoplasmic reticulum, and the Golgi apparatus. We have ribosomes here, the pink structures. And then we move into the nucleus. Down at the bottom here is a nuclear pore. So we move into the nucleus. And then the last panel is the nucleus itself, which of course is full of DNA that's all wound up in nucleosomes. But you can see there's no room to move around. Particles do not simply diffuse. Everything has to go on some kind of transport mechanism. And most of it is via these microtubules. As you know, they're kinesin and dynein motor proteins that bring things into the cell, towards the cell nucleus, and away from the nucleus by directed action. And that's how viruses, either free virus particles or viruses within endosomes, that's how they move around within the cell. They do not diffuse. So here's a summary of some of the different ways that viruses can enter cells after binding to a receptor. And it's at this point we're going to start talking about the destabilization of the virus so that it gives up its genome. Some viruses can actually enter right at the cell surface. So some envelope viruses, they fuse with the cell membrane right at the cell surface, and they deposit their nucleic acid right into the cell. Other particles bind to receptors and are taken up. This particular envelope virus has a icosahedral capsid. The envelope fuses at the surface, and then the capsid, it needs to get to the nucleus. It moves down the microtubules by dynein motors and eventually docks onto a nuclear pore. Here's another example of a virus that binds and enters by clathrin-dependent endocytosis, and the vesicles move into the cell towards the nucleus along, again, microtubules. And as we'll see, as these vesicles move towards the nucleus, they acidify, and that triggers destabilization of some virus particles. There are also other ways to get in the cell. There are clathrin and the clathrin-independent forms of endocytosis. There are caveolin-dependent forms of endocytosis. These are all different marker proteins that are found on the vesicles. Caveolin is kind of interesting because it targets the viruses to the endoplasmic reticulum. All right, so in all these cases, the viruses are moving around the cells either as particles or in vesicles by motor proteins. Now you have to remember, in addition, there's this microfilament network around the periphery of the cell, and that's not something you just walk through. You know, a virus can't just go through it. So it turns out that a lot of virus binding to receptors triggers signaling cascades that loosen up this microfilament structure so the viruses can get in. We'll talk a little bit about an example of that later. Now here's a movie of a vesicle, an endosome, moving along a microtubule using a dynein motor protein. Now it's a little bit humanized, I think, right? It looks like it's walking, but that's what they could think of to do. But it tells you what's going on is this protein is attached to the microfilament, the microtubule filament, and to the vesicle, and it moves the whole thing down towards the nucleus. So whether it be an endosome or a virus particle, these motor proteins are moving components around in the crowded cytoplasm. This, of course, is an energy-dependent reaction. You need to hydrolyze ATP or similar molecules in order for that to occur. This is produced by ex vivo scientific animation. Now, some viruses, as I mentioned, can fuse right at the plasma membrane. So the word fusion means two membranes come together and become one. And that happens a lot in virology because a lot of viruses are enveloped. And here are some examples of viruses that fuse at the plasma membrane. So at the very top is the overall scheme of what happens. The virus binds to its receptor, and then fusion occurs immediately at the plasma membrane. The virus and the cell membranes fuse. Then the nucleic acid of the virus comes into the cytoplasm, and then it can start to be expressed, which is a process we'll start to follow next time. Now, this is a bit unusual because fusion typically requires low pH, which happens in the endosome. But some viruses can do it at the cell surface because they have other ways of triggering it. One way is shown on the top panel of figures here. We have the viral membrane. Now we're looking very closely at the attachment of the virus to the cell membrane. In the viral membrane, we have two glycoproteins called HN and F. This happens to be the scheme for a family of viruses called the paramyxoviruses, including measles virus. The HN is the protein that will attach to the cell receptor. HN stands for hemagglutinin neuraminidase. And the other protein is called F. That contains the fusion peptide. As you may guess, that's the protein that's going to catalyze fusion of the virus in the cell membrane. Now what happens here is that when the virus HN attaches to a cell receptor, that's shown in the third panel, the red is the cell receptor. HN attaches to the cell receptor. That interaction triggers a conformational change in the F protein. So you see in the third panel here, the F protein is folded up near the virus membrane. The conformational change results in this protein unfolding, and part of it then sticks into the cell membrane. The part that's sticking in is the fusion peptide. This is a short 20, 30 amino acid sequence, highly hydrophobic residues that have the ability to poke into membranes. And normally this is hidden in the virus particle. So in the stable virus particle, this fusion protein is hidden. It's buried up by the virus membrane. Only when the virus attaches to the right receptor is this fusion protein exposed, so that it can now insert into the cell membrane and catalyze fusion. So this is becoming destabilized, and the catalyst here is binding to the right receptor. So as these viruses are floating around in your fluids, they don't want to be fusing with any membrane they bump into, so they keep the fusion peptide hidden until the virus binds the right receptor, which is presumably on the right cell, and then they can get in. So the fusion peptide enters the membrane and catalyzes fusion. How it does that we'll see a bit later for some other viruses. Now HIV also appears to enter at the cell surface, but it has a slightly different mechanism, and I show you this because it involves two receptors. Here is a schematic in the lower panel C of HIV, the viral membrane, the glycoprotein actually composed of two subunits, a transmembrane or TM portion, which is inserted into the viral membrane, and then the SU part, which is non-covalently associated with the TM. The SU binds the CD4 receptor. The CD4 receptor on the cell is shown in green. Upon binding to CD4, that exposes a high affinity binding site for a second cell receptor, which is a chemokine receptor, CCR, CCR4, CXCR5. And now we have high affinity binding of the SU protein to the chemokine receptor, and that second event triggers the exposure of the fusion peptide. So here's the fusion peptide on the TM in brown. It's that little squiggly bit at the tip. It remains tucked up against the virus until the two receptors are encountered, and only then is it exposed, leading to fusion. So both of these are examples of how you destabilize the virus. You're basically unloading the energy that's been put into this fusion peptide protein in order to catalyze fusion. So here's a movie of HIV infecting cells to illustrate this process. Here's HIV particles. They are red in the blood. The big purple guys are lymphocytes, CD4 positive T lymphocytes, which are the target of infection. These lymphocytes bear both receptors, CD4 and the chemokine receptor. Here's HIV coming in, and it has on its surface a glycoprotein. The SU part is shown in yellow. The TM is shown in blue. And the fusion peptide is folded up against the virus surface. We'll see that more clearly in a moment. So the virus is coming in, binding. OK, so CD4 is the blue guy, not the yellow one. Binds CD4, then high affinity binding to the chemokine receptor. And then what happens is the fusion peptide will unfold. The artists have removed everything for clarity. There's the fusion peptide unfolding. That unfolding is part of the destabilization of the virus particle, inserts in the membrane, and now the two membranes, the cell membrane at the bottom and the virus membrane, are going to be brought together. We're going to see how that happens in a moment. But for most viruses that catalyze this kind of fusion, it's by a hairpinning. The fusion protein continues to fold, and it brings the two membranes together. It turns out that all you need to do is bring membranes close enough to exclude water, and they will fuse, which is not easy to do. It requires energy. And the energy here is built into the F protein. That unfolding releases energy that does that. So here's a movie of this trimer of the glycoprotein of HIV. Here's the viral membrane at the bottom, and here's the cell membrane at the top. So the fusion peptide in this is already inserted into the cell membrane. And what we're going to look at is how this folding of this protein mediates the fusion of the two membranes. There's an association caused by a formation of another alpha helix there, as you can see, and that brings the viral and the cell membranes close to each other. So this is all as a trimer. The previous view was a monomer, and basically this unstructured area of the viral glycoprotein moves up and forms a second alpha helix, and as it does so, it brings the membranes together. You can see that illustrated here, and eventually they will get close enough in order to fuse. And at some point they bend a little to allow space, and there you see they're right together, the water is excluded, and the membranes can fuse. This is what I mean by hairpinning. The fusion peptides start out straight, they bend, and in so doing they bring the molecules together. And that's what we mean when we say the virus has become unstable. Here the glycoprotein is destabilized by the insertion of the fusion peptide in the membrane, which is catalyzed by receptor binding. All right, so that's repeating over and over again. So that when those membranes fuse, a pore is formed, and the viral nucleic acid can come out of the pore and go into the cell. So this question, which of the following does not play a role in virus entry? Clathrin-mediated endocytosis, fusion of viral and plasma membranes, diffusion of virus particles in the cytoplasm, microtubule-mediated transport, and lysosomes. All right, the answer here is C, of course, which does not play a role in entry, diffusion of virus particles in the cytoplasm. Remember, I said very clearly, viruses do not diffuse in the cytoplasm. They move around on motor proteins. So many of you got that right, but not all of you. Everything else plays a role. Clathrin-mediated endocytosis, fusion of viral and plasma membranes. And we showed that for a paramyxovirus and for HIV. Microtubule-mediated transport for sure. Lysosomes for sure. That's the endpoint of the endocytic pathway. So let's look at another virus in terms of how it fuses to get a little more detail on this hairpinning that I've been mentioning. This is influenza virus entry, which is probably the best understood in terms of how it works. Here the virus is bound to the cell surface via sialic acid, HA interactions. This causes uptake of the particle into an endosome. Nothing has happened yet. So it's not like on the cell surface the binding causes conformational changes. Here, conformational change requires acidification. And as part of the endosome pathway, as endosomes move from the plasma membrane into the interior, the endosomes have pumps in their membranes that pump protons into the interior of the endosome. That lowers the pH. So the pH goes from neutral to about 6 or 5.5, and that's enough to trigger a conformational change in the viral hemagglutinin. And that conformational change leads to fusion, and the viral RNAs can get out of the endosome into the cell. They want to actually go in the nucleus, which is shown happening here. That's an overview. Let's see what's happening in some molecular detail. Here, virus bound to sialic acid on the cell surface. We have two HA trimers shown here. The globular heads are binding sialic acid. As the virus moves into the endosome, the HA is still bound to sialic acid on the cell membrane. As the pH drops, the conformation of the HA undergoes a major change. And I can best show it to you by saying, if you look at the trimer of the HA, there's a very, very long extended alpha helix. Then there's a yellow portion that folds back, and then there's a shorter alpha helix down towards the base of the molecule. This becomes extended. In fact, this part of the molecule here extends up into the cell membrane, so the whole protein stretches out into an even longer alpha helical bundle. So now you have the fusion peptide exposed as a consequence of that. Normally the fusion peptide is buried down here. The fusion peptide inserts into the cell membrane. There are three of them. It's a trimer, so you see 1, 2, 3 in each case. And then this whole structure begins to fold. It hairpins just like the HIV glycoprotein does. You can see an intermediate here. And because the HA is attached to both the virus membrane by its transmembrane domains, and it's also attached to the cell membrane via the fusion peptides, as it bends, it brings the two membranes close to each other. Eventually, they fuse and a pore forms, and that's how the viral RNA can get out of the virus and into the cell. So this is a conformational change catalyzed by low pH, not by receptor interactions as we saw for the viruses that fuse at the cell surface. And again, I think if you look at the originals, you'll be able to trace where these fusion peptides come from. Here in this trimer, you can see at the very base, here's the part of the HA that goes into the virus membrane. There are 3 alpha helices here, at the tip of which are the fusion peptides. At low pH, these extend up and they insert into the cell membrane. The fusion peptides are shown in red here. And here's a view of just the HA molecule to show you what's going on here. These are X-ray crystallographic structures of 3 different forms of the HA molecule. This is a monomer. So here on the left is what we call an uncleaved HA. We see the long alpha helix composing the fibrous stem of the molecule, the globular head that attaches to sialic acid. This protein has to be cleaved by a cell protease in order for it to trigger fusion. The cleavage site is right down here at the base. And when this is cleaved, it exposes a new N-terminus, which is the fusion peptide. All right? So not only is the fusion peptide buried down here initially, like it is in those other viruses we talked about, but it can't be exposed unless it's cleaved. And this limits the cells that the virus can replicate in. That's one of the reasons why it remains in your lungs, because there is where the protease is that can make this cleavage. So on the left is the structure of the uncleaved HA, just to show you where the cleavage site is. Then on the right is the structure of the cleaved HA. Most of it looks exactly the same, except you can see here now there are new N and C termini, and the N terminus is where the fusion peptide is. That's going to go in the cell membrane. But so far it's still buried. The final trigger that moves this fusion peptide up towards the cell membrane is low pH. And on the right is the structure of the low pH form of the HA. Now the globular head is gone here. That didn't resolve in the crystallographic structures, but what you can see is that this portion of the molecule in green, which is relatively unstructured, consists of a few beta strands and loops. This is all extended to form more alpha helices. So here, from this point in the middle molecule on, this is all new alpha helix, and the fusion peptide would be at the top here. And that's what thrusts it in the cell membrane. At low pH, this reorientation and formation of additional alpha helices brings up the fusion peptide so it can insert into the cell membrane. So low pH is needed for this, and also cleavage. If the protein were not cleaved, it would not be able to reorient, and fusion would not occur. This is a type of fusion protein found in many different viruses. We call it a class 1 fusion protein. Here it is from influenza virus on the left, but you can see other viruses: simian virus 5, Ebolavirus, HIV, a retrovirus called Maloney murine leukemia virus. These all formed trimers. They are mostly alpha helical, as you can see by these structures, and they're perpendicular to the virus membrane. And that's why they look like spikes if you look at these proteins in the electron micrograph. So all of them work very much like the HA does in terms of reorienting a fusion peptide in order to achieve fusion of virus and host cell membranes. So this is another way of thinking of the destabilization of the virus. It's reorienting this protein, leading to fusion, and the virus essentially comes apart. But there have to be specific triggers for that. The HA has to be cleaved, and it has to be low pH in the case of influenza virus. So here's a movie of influenza virus entry. The virus is approaching the cell, binding to sialic acid receptors. It's taken up by the endocytic pathway. These are clathrin-coated endosomes now, which will move into the cell. And as they move in, of course, they become acidified. I think in a moment we're going to see them moving on a microtubule structure there, which is good. The clathrin comes off, which is part of the endocytic pathway. The vesicle, as it moves down, is becoming acidified. And there the endosome has fused with the viral membrane, and those are the viral RNAs coming out of the virus. They're going into the nucleus. That's the nucleus, this big structure here. They will go in the nuclear pores because this virus replicates its genome within the nucleus. So that's basically a summary in a pictorial form of what I told you. There's another class of fusion proteins which have different properties I want to tell you about. These are found in Dengue virus, which is shown here. They're also found in Zika virus and other viruses. These are flaviviruses. They're called class 2 fusion proteins. They're mostly beta sheets. Now the class 1 are mostly alpha helices, coiled alpha helices. These are mostly beta sheets. As you can see from the structure here, these are mostly beta strands forming beta sheets. They are typically dimers. So here is one copy, yellow, blue, red. That would be one copy. Yellow, blue, red would be the other copy. And they're parallel to the membrane. So you can see in this virus picture, that's a cryo-EM structure of dengue virus. You can see these dimers. So let's take a look at one right in the middle here. This green molecule, that's a dimer. There are two components to that. It's lying parallel to the membrane. And that's diagrammed here on the lower left. Here is the glycoprotein dimer lying parallel to the membrane. If this were influenza, they'd be sticking up. So these are class 2 glycoproteins. For the flavivirus, it's called the E glycoprotein. And these also catalyze fusion triggered by low pH. So on the bottom is the series of events leading to fusion. These viruses bind cell surface receptors. They're taken up in the endocytic pathway as the endosomes acidify. These proteins reorient. They stick up. Now, guess what's at the tip of these yellow domains? Fusion peptide, exactly. It's usually hidden here in its parallel conformation, but at low pH, this whole protein reorients. Fusion peptide sticks into the cell membrane. The glycoprotein hairpins. It begins to bend, and it bends, moves the membranes together, and eventually they fuse, and now there's a pore, and the virus contents can move out into the cytoplasm. So a similar mechanism, even though the protein, the glycoprotein, is parallel to the membrane, pH catalyzed fusion. The fusion protein is hidden, so the virus doesn't fuse with any membrane. and only ones in which it's being taken up into the endocytic pathway and the pH is dropping. So here's dengue virus entry into cells. There it's taken up into an endosome. It's moving into the cell along a microfilament, a microtubule structure. It's acidifying, of course, and we look inside the endosome. You're going to see how the viral glycoproteins stick up as the pH drops. Of course, they're going to insert into the endosome membrane. We're going to see a close-up of that now. Here, the reoriented glycoproteins, the fusion peptide at the top, sticking into the endosome membrane. Then they're going to hairpin, bring the two membranes together, and they're going to fuse. It's very insectoid, I think. I don't know why they drew it this way. Here are the two membranes fusing. They're fused, and the contents of the virus will come out. We'll see that from the exterior in a moment. There they go. And this is a plus-stranded RNA virus. So it's going to be translated right in the cytoplasm. That's where it has to go. Now, a couple of interesting twists on membrane fusion. Ebolavirus is really interesting because it turns out that the receptor that catalyzes fusion is in the endosome. So these are filamentous viruses. They're enveloped. They have an RNA protein nucleocapsid inside. They have an envelope with glycoproteins in the envelope. We think this is taken up into the cell by pinocytosis because no one's been able to identify a cell surface receptor for the virus. But at some point, the virus ends up in an endosome. And in the endosome, the viral glycoprotein, which are shown as these trimer structures on the surface, they're actually cleaved by enzymes in the endosome, late lysosome, to expose a binding site for a protein in the endosome called NPC1. And the glycoprotein then binds to NPC1, and that, together with low pH, catalyzes fusion of the viral and endosome membranes. So this is unusual because the receptor for fusion is in the endosome. So that gives it specificity for the right cell. NPC1 stands for Niemann-Pick cholesterol transporter. And there are people who are defective in the gene for this protein. They die about 10 or 12 years of age. They can't handle cholesterol balance properly. So that's why it's called Niemann-Pick disease. And this is the protein that's responsible. Defects in the Niemann-Pick cholesterol transporter gene lead to Niemann-Pick disease. So cells from these individuals are resistant to Ebolavirus infection because they don't have the NPC glycoprotein in the endosome. And that was important for figuring out what this receptor was for Ebolaviruses. Of course, if you have Niemann-Pick disease, it doesn't matter that you're resistant to Ebolavirus because you're not likely to get it anyway. And if you die by the time you're 12, it's not much of a consolation. But it could be that one day we can restore the NPC gene to these individuals by gene therapy. And that is a real possibility. Now, from all of this, what you should get is that fusion is regulated. It can't occur in the wrong location. Otherwise, the virus isn't going to put its nucleic acid in the right place. So sometimes it occurs at the plasma membrane, and that, of course, is at neutral pH. It's not acidified at all at the plasma membrane. So what catalyzes fusion is interaction with a second protein, receptor interaction, and then fusion protein is exposed. In some cases, fusion is triggered by low pH, but it's also regulated. It can be regulated by cleavage. Remember, I told you class 1 fusion proteins like the flu HA have to be cleaved to expose the fusion peptide. Otherwise, it's not going to fuse, so the right protease has to be present. I didn't tell you this, but for the class 2 fusion proteins, also cleavage is required, but of a second protein that allows the reorienting of that fusion protein containing glycoprotein. And for the Ebola viruses, there's an endosomal receptor for fusion. These are 3 different ways that fusion is regulated in the endosome. So obviously, it's very important to regulate it. Here's another way to look at it. Make sure the virus is destabilized in the right place, in the right cell, because you don't want them fusing randomly in your fluids if they're not near a cell that the genome is going to be able to productively replicate. So this whole idea that fusion is regulated is very important. Okay, the next question is, viral fusion peptides are exposed for insertion into the host cell membrane when the virus particle is near a cell, the virus particle is in the cytoplasm, trimers of the fusion peptides form, the endosome becomes acidified, or the virus is docked on the nuclear pore. So the answer is D, when the endosome becomes acidified. That's the only correct answer. When the virus particle is near a cell, not good enough. You have to attach to the right receptor on the cell. In the cytoplasm, well, by then fusion has already occurred if the virus is actually in the cytoplasm. Trimers form. Trimer formation itself does not catalyze fusion. Remember, you have to have cleavage of the HA and low pH as one example. And the virus is docked on the nuclear pore. Those viruses are icosahedral protein-based viruses. They don't undergo fusion, like adenovirus. And we will talk about that in a moment. So let's talk about how adenovirus gets into cells. So adeno is not a membrane envelope virus. How do the nucleic acids get out? We talked about how these icosahedral particles bind receptors. But how do you destabilize the particle to get the genome out? We talked about that for envelope viruses. It all involves fusion and regulated fusion. How do these icosahedral viruses work? So let's look at a couple of examples. Here's adenovirus. Binds to two different cell surface receptors. It's internalized by clathrin-dependent endocytosis. As the endosome acidifies, the virus particle begins to break up. Not completely, it remains intact, but a protein that's on the interior of the capsid, shown as these little diamonds here, gets released. And that protein pokes holes in the endosome membrane. It's a porin, it forms pores. Now normally it's buried in the capsid, so it doesn't poke holes in any membrane that the virus might encounter. But at low pH in the endosome, after getting into the right cell by receptor interaction, the low pH releases that protein, it pokes a hole in the endosome. The particle, what's left of the particle then gets out. It attaches to a dynein motor on a microtubule and gets transported to the nuclear pore, docks onto it, and the genome comes out. So the particle by this time is degraded enough so that the genome can get out, but it's not degraded too much so that it can't be recognized by the dynein motors that bring it to the nucleus. So there you go, endosome-mediated partial disassembly and escape from the endosome. Here in these electron micrographs, really nice pictures, you can see an adenovirus particle traveling on a microtubule. You can even see the motor protein attached to it. And then at the bottom here are two adenovirus particles docked onto the nuclear pore. So the nucleus is at the top of the image, cytoplasm at the bottom, and these are two nuclear pore complexes. Poliovirus is a different example. So here we have this lovely shell, really stable. How do you destabilize it? In this case, it turns out that all you need is the receptor. All you have to do is bind the receptor to this particle, and out will come the nucleic acid. So here we have poliovirus binding to its receptor on the cell surface, gets taken up by endosomes, but very early on, the RNA comes shooting out of the particle because the receptor destabilizes it and leads to the formation of a pore. It induces major structural changes in the capsid, which opens holes up, and the RNA snakes out of that. It must also make some kind of a pore in the membrane. And that's diagrammed at the bottom here. Here's a close-up view of the virus particle in panel B, bound to two receptor molecules on the cell surface. The binding of the receptor induces major structural changes in the particle such that a pore opens up at the five-fold axis of symmetry or somewhere nearby, and the RNA comes out. Where exactly this pore isn't clear. I'm showing it as the five-fold axis, but it could very well be somewhere else in the particle. Now, part of this pore is formed by viral proteins shown in blue. These are normally hidden in the virus particle. They're VP1. They're the N-terminus of VP1. They're hydrophobic. And upon binding the receptor, they come outside, insert into the cell membrane, and make a pore. So this is kind of like HA-mediated fusion, except we don't need acidification in the endosome. All you need is the receptor. The receptor pops onto the virus, pulls out the N-terminus of VP1 to make a channel in the membrane, and makes a hole in the particle so that the RNA can get out. So here, the destabilization of polio is due to the receptor. And if the virus bumps into the wrong cell that doesn't have a receptor, it's not going to give up its genome. Here's a close-up look of what's exactly going on in poliovirus when this is happening. On the top is a diagram of the receptor binding into that groove around the five-fold axis. Turns out that there is a little tunnel just below where the receptor binds, and in all of these polio-like viruses, it has a little lipid in it. And one of these is called sphingosine. They're carbon chains of 16 or so amino acids, but they're lipids. They fit in there very tightly. And when the receptor binds, the lipids get forced out. That leaves a big hole, and that allows the proteins of the virus to reorient and make that channel through which the RNA comes. Now here's the lipid shown here in this image at the lower left. That's one of the subunits of the particle. Here's where the receptor would be binding in this little groove. It's the five-fold axis of symmetry. The receptor sits down here in the groove, the lipid goes out, and the virus reorients. Now, how do we know this? We know this because there is an antiviral drug that displaces the lipid in the capsid, and when the receptor sits down on it, it does not leave. So the virus cannot uncoat. That blocks infectivity. This is a structure of the drug, one of these drugs fitting into this pocket. So again, there's the little pocket underneath the receptor. The drug substitutes for the lipid. When that virus now attaches to a receptor, the drug doesn't leave. It's locked in because it's binding with very high affinity, and that ends the infectious cycle. So that's an example of how an antiviral drug can work. On the bottom is the structure of polio, complex with this drug. You can see them in yellow. And of course, there are 5 around each five-fold axis of symmetry and 60 for each molecule. Now, this drug never made it to the clinic because there was too much resistance emerging in trials, because it only takes one amino acid change in the capsid protein surrounding this drug to make resistance. But it did illuminate how uncoating of the virus works. Let me give you an example of why you would need 2 receptors for a virus to get in cells. We talked about HIV, where you need 2 receptors for high affinity interaction and exposure of the fusion peptide. Here's an example with the Coxsackie virus receptor. These viruses need 2 receptors. One is DAF, decay accelerating factor, and the second is CAR, the Coxsackie adenovirus receptor. Now, Coxsackie viruses initiate infections at epithelial surfaces, either in your respiratory tract or your gut. CAR is a component of tight junctions, though. How does the virus get to it if it's in a tight junction? DAF is on the cell surface, but not CAR. Well, it turns out that the viruses initially bind DAF on the apical surface of polarized epithelial cells. That binding starts a whole signal transduction pathway, which is shown here by these arrows, ABL, RAC, FIN, various protein kinases that do 2 things. First, they loosen up the actin filaments just below the plasma membrane. And you need to do that because what's going to happen is the virus bound to DAF is going to move to the junction. The second thing that this signaling does, again, the virus binds to DAF, initiates a signaling cascade, loosens up the actin filaments, and it also loosens up the junctions between cells. And that allows the virus DAF complex to move into the junction, bind to CAR, and get into the cell. So that's why this virus needs 2 receptors. One to bind it to the cell surface, that's DAF, and CAR allows it to get in. DAF alone isn't enough. The virus can't be endocytosed from the top, doesn't work through DAF, but CAR does, but the virus needs to get to CAR, and that's where DAF binding gets in. Really brilliant, I think. So reovirus is another interesting example. I kind of highlighted this last time because this is a virus with 2 concentric icosahedral shells. And I told you I would explain to you why we think it has 2 shells, and that's shown on this slide. And these viruses, shown in the upper left, 2 shells, an outer shell and an inner shell in 2 different colors. They have 2 different triangulation numbers. They bind to receptors on the cell surface. They're taken up by receptor-mediated endocytosis. And then as these endosomes move in, eventually they fuse with lysosomes, and they pick up all the enzymes that are in lysosomes. Now, most viruses get out of the endocytic pathway before they hit the lysosome because that's a degradative site, and most viruses will be digested by all the proteases and nucleases in lysosomes. But these viruses are unusual. They stay in as far as the lysosome because they need those proteases to become uncoated. So what happens is the outer layer of the capsid is stripped off by the proteases that are delivered to the endosome by the lysosome. And first, the outer proteins are taken off. You have the virus particle that comes in. You then have an intermediate subviral particle. And then finally, you have what's called a core. So the entire outer surface is stripped off. And in so doing, it exposes hydrophobic proteins on the inner core. And that allows the virus to penetrate out from the lysosome into the cytoplasm. So again, we've talked about a receptor destabilizing the virus. We've talked about low pH destabilizing. Now a virus that needs proteases from the host cell in order to be destabilized. And that's why there are two shells. The outer shell, it needs, when it's traveling among hosts, it's very protective. The inner shell has got the hydrophobic residues that allows the virus to get out of the endosome. So we've looked at a couple of different ways of viruses getting from the surface into the cell. Let's now end up talking about how some viruses get in the nucleus, because not all viruses do that. Many RNA viruses stay in the cytoplasm, but most DNA viruses get in the nucleus, and there's a special set of problems there. The nucleus, of course, is a double membraned organelle, and things just can't get in. They could go through nuclear pores, which are very specific in what they will let in. There's a size limit to nuclear pores, and beyond it, you cannot get in. Of course, getting into the nucleus via the pore requires a whole transport system, which is energy dependent and so forth, and only certain viruses can get in without special tricks. So here on the left is influenza virus. The endosome is just fused with the virus membrane, and all the viral RNAs are now out into the cytoplasm. And these viruses, remember, have 8 RNA protein complexes in them. They need to get in the nucleus. They're actually small enough to go through the nuclear pore. And they use the transport system. There is a nuclear localization signal on the proteins on the viral RNAs. That gets them into the nucleus. Most other viruses are too big to get in the nucleus. So here we have an example. This is a herpes virus docking onto the nuclear pore, and the DNA coming out of the portal. Remember, in herpes virus capsids, one of the 5-fold axes is a hole or a portal that can take the DNA out. So these viruses dock onto the nuclear complex, and the DNA goes inside. We talked about how adenoviruses are partially disassembled. They're still able to dock onto the nuclear pore, and then active transport mechanisms pull the DNA inside of the cell. And then finally, parvoviruses, these are very small. They seem to bind to the nuclear pore, and that induces somehow permeability elsewhere in the membrane, and then the virus goes through there. And in the nucleus, they uncoat their genomes. So getting in the nucleus is a special problem. I want to leave you with a little detailed examination of how adenovirus puts its DNA in the nucleus. Remember, adenoviruses get partially disassembled in the lysosome, in the endosome. They come out into the cytosol, they bind microtubules, and then the dynein motors bring them down to the nuclear pore. They then dock on the nuclear pore, you can see here. We think that the particles remain attached to the microtubules, and then the kinesin motors start to pull them away. But they're docked very tightly onto the pore, so as the motor pulls them away, it breaks up capsid, and that allows the DNA to get into cells. The DNA is actually coated with proteins, and we think these are recognized by the nuclear import system. Here's the remnant of the broken capsid. So it's kind of an interesting idea, right? It's the particle is docked on the pore, and then as the motor tries to pull it away, because that's its natural function of the kinesin motor, it breaks up the capsid and allows the DNA to get into the cell. So that's an example of utilizing cellular pathways to get your DNA into the cell.